So I will be talking about quinoa as a climate resilient crop as a success story for Pakistan. Before going into the details of my experiment, I would like to show you a brief documentary about the background of the study.
In the present scenario of climate change and increasing population, use of climate resilient alternate crops for food can impart to food security and health benefits. Quinoa is one such superfood plant with various medicinal and nutritional benefits. It is highly resistant to harsh climates and soil conditions. It is consumed by people as a whole plant with high nutritional value. Used as rich nutritional source, it's feeding from animals such as cattle, pigs, and poultry. Proteins content of quinoa seeds varies from 13 to 16%. The total protein content of quinoa is higher than that of rice, barley, corn, rye, and sorghum. The main carbohydrate components of quinoa is starch, and it contains 52% to 69% of it. Quinoa is also accepted as an alternative oily seed. Quinoa is very beneficial in various prevent disorders. Antioxidant activity of quinoa helps in immune regulations. Soil salinity is a major environmental problem that is interrupting more than 20% of the world irrigated areas. Quinoa is one of the most promising examples of plants which are extremely tolerant against salinity. Dr. Ghulam Abbas is working on quinoa in Pakistan. He has collected and introduced quinoa germplasm from different countries of Europe, Australia, and USA, and succeeded to screen out four best genotypes with high yield potential under harsh climatic and soil conditions like heat, salinity, and drought in Pakistan. We further reported that quinoa is the best food crop for arid and semi-arid regions in Pakistan.
Thank you for listening to this brief documentary. So, oh, how to move to next? Okay. So let's start with the contents of the presentation. I will start with the introduction that has been covered in the documentary. Then I will go to the objectives and briefly methodology, result, and conclusion. As it has been highlighted in the documentary that climate change is a very dangerous issue in recent times, particularly for arid and semi-arid climatic conditions. Those are prevailing in subcontinent like Pakistan, India, and Bangladesh. So in Pakistan, a lot of area is salinized, that is having soil salinity and sodicity issues, accompanied by drought. And in recent times, we are facing heat stress as well. So in the past, we tried to get some benefits out of the salinized soil by doing some physiochemical and some other practices like reclamation of salt affected soils or using good quality irrigation water. But those practices were not up to the mark. So coming to the recent years, we are left with the introduction of new crops, actually, like quinoa. Quinoa is not a native crop for Pakistan. So we have obtained some germplasm of this quinoa from different countries of the world. For example, from Denmark, Australia, and its basic origin that is Latin America. So comparing quinoa as a superfood or highly nutritious food with other cereal grain, you can say that it is having a very high content of proteins, and its carbohydrate contents and energy contents are also comparable with other cereals such as rice, barley, wheat, corn, rye, and sorghum. So that is another important aspect of quinoa. That is, it is not only suitable for saline and water limited areas, but at the same time, it is having very high nutritionist value as well. So this is an overview of the nutritious value of the quinoa. In addition to previous contents, it is also having a very high profile of minerals, and it is also rich in multivitamins. So that's why it is also called as superfood. And at the same time, it is also called as miraculous plant due to its high nutritional value and its high resistance against multiple abiotic stresses. So this is brief overview of the salinity problem that is actually happening in Pakistan. We are having approximately 20% of the area that is highly salt affected soil in Pakistan, and salt content may go up to 40 millimole per liter, or you can say that is around 40 deci per meter. It is really high content of the soil. So this is brief about the salt tolerance mechanism of quinoa: osmotic adjustment, organic osmolytes accumulation, antioxidant activities, epidermal bladder cells, and ionic homeostasis. Similarly, temperature stress is also increasing with time due to climatic change issues, and the optimal temperature for quinoa, that is around 20 degrees centigrade. It grows best between 15 to 20. But in Pakistan, we are facing temperature as high as, you can say, 45 Celsius. So it is becoming very challenges to grow our native crops, for example, wheat and rice, who can't go beyond even 35 degrees centigrade. Similarly, as it has been stated that quinoa is resistant against salinity and high temperature. Similarly, it is also a drought tolerant crop as well. So we are having very low quality water in Indo-Gigantic issues. There are a lot of salt precipitation, and in addition to soil salinity, we are having water salinity as well. So it would be a better option if we can grow crops which require minimum amount of water. So quinoa is again such a very good option. So this is overview of quinoa as a climate proof crop because it is tolerant against salinity, drought, and against heat as well. But that issue is tolerance against high temperature or heat stress that needs a bit further investigation as well, because this aspect has not been explored so far. So aim and objective of the study, as it was briefly described in the documentary, that quinoa is not native to Pakistan. So my team in Pakistan has tried to obtain the germplasm of quinoa from different countries of the world, and we are trying to introduce those lines or varieties in our country. So we conducted different types of experiments under controlled conditions in glass houses, and from 8 to 10 genotypes, those were collected from different countries. Now we are successful to have two to three genotypes. Those are better adopted under field conditions against salinity, drought, and heat stress. So this is a brief about the soil and water characteristics. As you can say, the EC of the soil, that is quite high, 11, more than 10 EC in terms of deci per meter. At that much EC, most of the crops, particularly glycophytes like maize, those are not able to survive. However, we can grow wheat with approximately 50% reduction in yield. So this is not the maximum level of salinity that we are having in Pakistan. That is of that soil from where we get the sampling and where we grow the crop actually. As it was shown in the previous style, we are having the soil where EC level is up to even 30 deci per meter. So on those soils, we are not able to grow any type of crop over there. So we are left with the only option to introduce new crops in our cropping system to get some sort of benefit on those soils. So this is overview of our pot culture experiment. In this experiment, initially we grew approximately 8 to 10 different genotypes, and under different salinity, water, and drought stress levels, accompanied with heat stress, we were able to isolate two genotypes. Those were further grown under field conditions. Those two genotypes were Puno and Titicaca. These both are having Danish origin. These are belong to Denmark, both of these genotypes. So we conducted field experimentation during the last 2 or 3 years in district Vehari, that is located in Punjab province of Pakistan, where we are having a large area of salt-affected soils. In addition to salinity, drought and high temperature is also prevailing over there. As you can see, the average rainfall that is less than 125 millimeter. So this is an arid climatic condition where we are having very less amount of rain and very high evapotranspiration. Due to that, the salt content in the upper layer of the soils that is quite higher. So we grew these both genotypes on the natural field conditions, salt-affected soils, with a gap of 10 days. We sown the genotypes 3 times during the month of November. The objective was that the late sown genotypes will have to face high temperature at their post-anthesis, or you can say at their grain filling stage. That is the most susceptible or sensitive stage of quinoa against high temperature. So this is the temperature variation during various phenological phases of quinoa. As you can see, starting from emergence, leaf formation, panicle, flowering, fruiting, maturity, and harvesting. By maintaining the difference of 10 days, we were able to have high temperature at post-anthesis stage in case of late sown genotypes. So we followed the recommended plant cultivation practices as those has been reported by previous researchers. Plants were irrigated using the natural source of water, that was saline water actually. And for water stress, water was not applied to the plants after post-anthesis stage. And we took the plant up to the final harvesting stage, where we recorded grain yield and other quality parameters. So this is an overview of our field experiment. You can see from the slides, this whitish color actually, that is an indication of highly saline areas, high salt content in the soils. So these are brief about the results. These parameters were taken at the vegetative stage, chlorophyll content and stomatal conductance. So as you can see, both of these parameters are decreasing if we go from D1, D2 to D3. D1 actually that is early sowing and D3 is late sowing. In case of late sowing, due to heat stress accompanied by the soil salinity and drought, the physiological parameters were decreasing in a linear fashion. And if you compare both genotypes, Puno and Titicaca, the performance of Puno was better as compared to Titicaca. Same was the case in case of sodium and potassium uptake. Potassium uptake was decreased and sodium uptake was increased with the increase in salt content accompanied with drought, and particularly then both stresses were combined with late sowing. Oxidative stress, you know that under abiotic or even biotic stress conditions, there is a production of reactive oxygen species in plants. So this is an indication of the oxidative stress in plants. H2O2 content and TBA contents, those were increased. And in order to overcome these stresses, the activities of different antioxidant enzymes, those were also increased in a linear fashion. So again, the activities of antioxidant enzymes was the highest in case of D3, and particularly that was higher in case of Puno as compared to Titicaca. So again, this is an indication of the higher stress, where all the three stresses were combined together. salinity, drought, and high temperature. So this is the grain maturation, or you can say the final harvesting stage of the plants. Plants were harvested at this stage. Then we recorded plant growth and green yield parameters, plant biomass, grain yield, harvest index, and 1,000 grain weights. So again, these parameters were higher in case of Punno, where only the salinity was applied. But when salinity was applied with drought, particularly under late sown or heat stress conditions, plant biomass and grain yield was decreased. And minimum grain yield was obtained in case of Titicaca as compared to Punno. So Punno performed better as compared to Titicaca. Grain mineral contents, they also followed the same trend. Grain quality attributes like protein, carbohydrates, fats, and fiber. Same trend was recorded in case of these parameters. So the conclusion is that due to climate change issues, the problems like soil salinization, drought, and heat stress, those are also increasing under arid climatic condition of Pakistan. So in addition to having better genotypes of our pre-existing varieties, we need to have new germplasm in our system. So that's why we are working on introducing halophytic crops such as quinoa in our country. And the results of this study indicated that among different germplasm collected, Punno and Titica, these two performed better. But when they were compared under real problematic condition, under field conditions, Punno performed better as compared to Titicaca. So that is all about my presentation. And I am highly thankful to the Higher Education Commission of Pakistan for providing research funds, and Comsats University Islamabad for providing research facilities, and UC Dublin for providing this platform to present my research over here. Thank you so much.
Research, but again, kind of we're falling back behind further. So I think again, we'll leave the discussion for the lunch break.
Okay.